is an obligate intracellular parasite, something that can only reproduce inside a cell. Nutritionally, viruses would fall into that category. But they're not organisms because they're not cellular. On the other hand, they can't reproduce anywhere but inside a cell, so I suppose technically they're hypertrophs. They get into all kinds of organisms. Probably every other type of organism has been infected by viruses. So they're not unusual. And if you really want to think about what viruses are for, my suspicion is that viruses are like UPS service or postal service. They're an exchange, they're an information exchange mechanism. They allow for exchanging nucleic acid between organisms, which kind of begs the question, where do they come from in the first place? But we really don't know. Size-wise, viruses are much smaller than cells. As soon as I say that, I think, oh, even there's some exceptions. Typically, viruses are much, much smaller than cells. They vary considerably, and you can find lots of images of viruses online on the web. You can find electron photomicrographs, and you can find illustrations of viruses. You can find models of viruses. We have our own model of a virus in here that was built by a student. For a long time, how viruses were going to be categorized was subject to change. But because viruses were initially found within living organisms, people that were studying animals ended up studying animal viruses. People that were studying plants ended up studying plant viruses. People studying bacteria ended up studying bacterial viruses or bacteriophage. And there wasn't a consistent single group of viruses. That's beginning to change. Exactly how these things are recognized and categorized is also changing. We'll get into that a little bit. When you're talking about a complete virus particle, and that's what this is a model of, this is a complete virus particle. This complete virus particle, as it exists outside of its host cell, is called a virion. So last time I was talking about viroids, I said watch out for spelling, kind of like when I said watch out for spelling of nucleus and nucleoid and nucleosome and so forth and so on. So a virion is a complete form of a virus. It's found outside the cell. This is the infected form of a virus. And last time when I was talking about transduction, which is genetic exchange in fallen viruses, what I said is if this virus, this is a model of a bacteriophage, when this thing actually infects the host cell, the only thing that goes in is the nucleic acid, this part. But in order to be an infected entity, it has to have this complete structure. So, whoops, I need to put it down so it doesn't take. So this whole thing is a virion. And again, lots of different forms. You can find images of this thing. You can find electron photomicrographs that are a little bit difficult to really see the exact structure. And so there's lots of models and illustrations that are more specific. As far as where did viruses come from, this is somewhat unclear. I'm going to give you an example of a virus type that infects Escherichia coli. The virus type is called bacteriophage lambda. And there's kind of an interesting scenario by which bacteriophage lambda can enter the host cell chromosome. So the question is, could viral nucleic acid be something that came from cells in the first place? Or did viruses evolve separately from cells and then get in? We really don't know. The study of viruses is virology. As sciences go, this one is younger than microbiology because microorganisms were visible before viruses were visible. These things could not be visualized at all without electron microscopy. And electron microscopy was more slow to develop than other types of microscopy. You can't see these with a light microscope. Consequently, when we're working with viruses in the laboratory, which we will, all we can see is evidence of virus infection. We can't see the viruses themselves. We can see where they're killing bacteria. If you were working with animal viruses, you typically grow them in tissue culture, and you can see where they're killing other types of cells. But we're not going to be doing that either. So this is kind of a recall, only in the sense that I mentioned it when talking about transduction. the term bacteriophage applies to a virus that infects bacteria. And as I said, that word means bacteria eater. They don't actually eat bacteria, they kill them. And so evidence of bacteriophage infection leaves a little clear spot in a bacterial lawn. It's called a plaque, sometimes called a window, but it's actually a little divot where the bacteria are missing. If a virus infects Escherichia coli, it's called a colophage. Probably the reason that colophage are so recognized is because Escherichia coli is thoroughly studied and it's used in lots of experiments. So lots of people have worked with colophage. And most of the best known viruses are colophage. So again, virions can vary considerably in size and shape, but they have specific structures that we can talk about, things that they have in common. And a lot of what I'm going to tell you about bacteriophage is also applicable to viruses that infect other types of organisms. And I think about, there's all those other microbes, protozoa and algae and so forth, fungi. How much do we know about the viruses that infect those guys? I don't know. They're not separate groups usually. Anyway, a virion always has a nucleic acid core. As I mentioned here, it's the purple pipe cleaner, that nucleic acid core. The nucleic acid core is always surrounded by a protein coat called a capsid. So we want to know some more detail about that nucleic acid core. And again, don't confuse virions with viroids. Virion, again, is complete virus particle as it exists outside the cell. A viroid is naked RNA, not a virus at all. So we've seen the term genome applied to the total DNA content of the chromosomes within an organism. Well, we can also apply that term to the DNA content within a virus, within a virion. The genome, it's the total DNA content, again, usually DNA or RNA, occasionally both. There are some other aspects of the genome that I want to give you some specifics about what kind of structure they can have. This little exception that's mentioned here, there is a virus called mimivirus. It's a virus that infects acanthamoeba, and it contains some messenger RNA molecules. It's a DNA type virus that has RNA. So they've actually found viruses that have both DNA and RNA. Rather rare. But how else does the nucleic acid core differ? Well, as I said, DNA or RNA. Cellular nucleic acid is both, but the genetic information, the storage component, is DNA. Viruses, the storage component can be RNA. If it's all they've got, that's what they're going to have. We learned that DNA is a double-stranded molecule and RNA in cells is single-stranded, although it can wrap back on itself. So transfer RNA molecules wrap back and they have regions that are double-stranded. In viruses, DNA can either be double-stranded or single-stranded. And likewise, RNA can be either double-stranded or single-stranded. If the viral nucleic acid is double-stranded RNA, the strands are complementary to one another and anti-parallel. So they follow the same rules that DNA follows, they just have different nucleotides. They have RNA-type nucleotides instead of DNA. We learned about differences between eukaryotic and prokaryotic cells. I said typically eukaryotic cells have linear nucleic acid, whereas prokaryotic cells mostly have covalently closed circular DNA. Turns out they don't all. Bacteria in the genus Streptomyces have linear DNA. They may even have linear plasmids, which is pretty frustrating. But viruses can have both. Different types of viruses infecting different types of organisms, they can either have linear nucleic acid or they can have covalently closed. There's no consistency within viruses. The other thing that's different about these is how big. Typically, prokaryotic genome is much, much smaller than eukaryotic genome. Viruses vary considerably. I think the smallest ones have about 4 genes. The ones that have larger genomes can have 400 or more. So they can have pretty large sized genomes or they can be really little. And again, prokaryotic cells, only one chromosome, usually. That's not entirely true. Eukaryotic cells always have 2 or more. Viruses can have 1 or they can have many. And again, they range considerably in size. Okay, so that's the genome, the nucleic acid content. A lot of different ways that these can vary. Next, we're going to look at the protein coat, which is called a capsid. That capsid on this model is this structure that you see that has these triangular portions on it. A lot of capsids are polyhedrons of some sort, but they aren't all. If they are polyhedrons, then they're made up in smaller units called capsomeres. Again, remember I said polymer, monomer, dimer. Capsomere just means a capsid unit, and they're oftentimes triangular, but they vary quite a bit in shape. Here's some illustrations of viruses or models of viruses. These things are really quite complex because the capsomeres themselves are made up of protein complexes. Here you see a diagrammatic representation. The capsid is this thing that looks like it's made with a bunch of triangles, and the individual little balls are the capsomeres. But capsomeres themselves are made up of still smaller units called protomers. Now, capsomeres are not visible. Well, you can't see a virus with a light microscope anyway, but capsomeres are just barely visible with electron microscopy. They appear to be triangular in shape, making viruses, even if they look like they're spheres, they're actually a bunch of little triangles. They're polyhedrons. So here's another example. This one has a tendency to try to fall apart. But here you see what looks like a spherical structure, more or less. But then you look closely at it, it's a polyhedron, a whole bunch of little triangles. These little triangles then would be the capsomeres, and each one of these capsomeres is made up of still more complex protein structure. So it becomes more and more complex. Capsomeres are made up of protomers. These are just protein complexes. Now this is a linear type virus, and so here the capsid, which is this long tubular looking structure, is made up of capsomeres that are circular. So this one doesn't have the triangular shaped capsomeres, but a lot of viruses do. And again, their shape is quite variable. Now, in addition to having a nucleic acid core and a protein coat, this capsid, some viruses have another layer outside, which is called an envelope. If they have an envelope, it's because they stole it from a host cell. Envelope has basically the structure of cell membrane, so it's typically a lipid bilayer with peripheral and integral proteins. The virus can't make something like that, but it takes that with it as it exits its host. So it pulls a piece of this host cell membrane, and that's what forms the envelope. A lot of times there will be proteins on the outside that are encoded by viral genes. So this is a model of HIV, which is an animal virus. These structures that are on the outside, these are proteins encoded by viral nucleic acid, but then they have moved to the surface of the cell, become incorporated into the cell membrane, and then when this virus exits the cell, it takes that membrane with it, takes pieces with it, which leaves the host cell with a lot of holes in it, and typically that's why the host cell dies. But not all viruses have envelopes. Some do, some don't. The other thing not all viruses have is a tail assembly. This particular model has a rather elaborate tail assembly, and the name is appropriate because this is a protein structure that is assembled. It's put together, so it's built inside of a host cell. This particular tail assembly has a collar, a tail that has an outside sheath, and inside a tail core. Then there's a base plate. Then there are these things that look like legs. These are called tail fibers. And underneath, these little things that stick down are called tail pins. So here you see again the collar, the tail, which has a tail core and a tail sheath, a base plate with a bunch of pins, and the little tail fibers sticking out from that. Again, there's a lot of variation because that one shows a collar with little protein pins coming off of that. They don't always have that. But again, not all viruses have the tail assembly. Regardless of whether they are DNA or RNA, whether they have an envelope or not, whether they have a tail assembly or not, typically viruses all have the same strategy. The strategy of a virus is, number one, get inside a host. Number two, reproduce. Number 3, get out. That's all they do, is get in a host, reproduce themselves, and get out. So gain access, we'll talk about how they can get in, reproduce inside that host, which is a mechanism completely unlike any cell reproduction process we've talked about. And then get out, escape. They just do this over and over again. The effect on the cell is variable because some viruses are cytolytic. If they kill the host cell, then depending on what cells they're killing and what kind of a host we're talking about, the damage may be significant or not. If they get into a bacterial cell and kill it, well, that's the end of that cell. If they get into a multicellular organism, they might be able to kill some cells without causing a tremendous amount of damage. It just depends. Anyway, here we see again this diagram. You can find lots of different illustrations of viruses. So you see what's sometimes referred to as the head. Technically, this region up at the top is called a nucleocapsid. So it's the capsid plus the nucleic acid core inside. So for this guy, this is the nucleocapsid, and everything from here down is tail assembly. They always have to have a nucleocapsid. They don't necessarily have a tail. Now, typically when a virus gets into a host cell, that virus, actually what gets in is the viral nucleic acid. And that viral nucleic acid is going to somehow disrupt cell function and take it over so that the cell then is going to reproduce viral parts. Sometimes it causes the lysis of the cell, sometimes it doesn't. We'll talk about a cytolytic pathway, and we'll talk about what's not cytolytic, which is often referred to as a temperate phage. And again, if it gets into a multicellular organism, the amount of damage caused depends on how many cells are infected and what kind of cells are infected. If it's surface cells, say skin cells, epithelial cells, there can be minor damage, maybe a rash, maybe a painful sore, that then heals up and you don't notice that the virus isn't about anymore. Although human herpes viruses are what we call latent viruses, so once you're exposed to them, you have them. They stay with you for life. And they can reactivate at another time. Bacteriophages, because they're easy to work with, primarily because they live in bacteria and bacteria are easy to work with, we know a lot about how they work, and they've been used to study life cycles of viruses, how these things work. Also, you can work with bacteriophage in a laboratory like this. You don't have to worry about being infected by them. I suppose you might have to worry about them infecting your E. coli. So in the lab, not tonight, but next week, we're going to do exercises with bacteriophage. Again, you don't get to see them, but you just get to see the effect they have on bacterial populations. You see the results of virus activity, and they're not damaging to you in any way. So the other terms, we've used the term in vitro. When we grow organisms in artificial containers, we grow them in vitro. You cannot grow viruses in vitro unless you're growing them inside bacteria that are grown in vitro. Viruses are always grown in vivo, inside living organisms. So we grow them in vivo in bacteria that are grown in vitro. So again, we're going to talk about two different types of life cycles for viruses, cytolytic viruses versus temperate or avirulent. So we'll talk about the lytic ones first. Cytolytic bacteriophage. The term cytolytic, cyto means cell, lytic means to lyse. So a cytolytic virus is one that's going to ultimately cause cell lysis. The cell's going to blow up, it's going to be destroyed, and release a bunch of new viruses when it does. Again, lots of different images that can be, the one in the middle is an illustration. The two on the outside are actually electron photomicrographs, but the one on the right is artificially colored. So you can see the virus particles. So when they attach to a cell surface, you can have lots of viruses attaching, and the nucleic acid is going to get inside. A phage that is called temperate or avirulent or asymptomatic would be the type of virus that does not kill its host. And then I have to qualify that and say, well, they don't kill them right away. And that's because viruses can switch back and forth. They can be cytolytic and then they can be temperate. They go back and forth. They have to be able to do that if they're going to be involved in transduction. So last time I talked about transduction, how a virus can either be cytolytic to start with or a prophage to start with, but it has to end up being a prophage at the end, otherwise transduction hasn't been accomplished. And last time we learned the term prophage. The term prophage refers to viral nucleic acid that has been incorporated into the chromosome of a host cell. If it's a prophage, it has to be DNA, because the host cell chromosomes are DNA. So whether we're talking bacteriophage or animal virus, plant virus, whatever, actually the term prophage only applies to bacteriophage. If it's an animal virus, it's called a provirus if it goes into the chromosome. So this particular model represents a group of bacteriophage called the T-even bacteriophage. These are colophage. So you'll see lots of images of these things online. They've been very thoroughly studied because they infect Escherichia coli, and E. coli is really easy to grow in vitro. So it's easy to grow those guys and to see what kinds of things are going on with their viruses. We're going to look at a cytolytic life cycle involving a T-even phage. So the life cycle begins with a virion, a free virion in the environment. If this model of a virus represents a free virion, it's very much too large to be infecting an E. coli cell if the E. coli cell was the size of this front bench. So you saw the electron photomicrographs. If this thing was more actual virus size, it might be about this big for the front bench. And you have a whole bunch of these guys infecting this bench. So this guy's quite a bit larger. That gives you an idea of how small these guys are. Again, the virion is the complete form of the virus, so you have all the components of the virus. This thing is going to attach to the surface of the host cell. This attachment step, for a long time, references called it adsorption. Adsorb means sticking something to a surface. Now a lot of the references are saying, oh, we'll just use the word attachment. So you can call that first step either adsorption or attachment. But in the case of a phage like this involves a very specific interaction between components of the virus and the host cell surface. So this particular virus, again a T-even phage, has receptor sites at the ends of its tail fibers. And those receptors have to make contact with the host cell surface. This one has 6 receptor sites. They all have to be attached to a specific receptor on the host cell surface. And so this thing has some capability to move around until all of those have made contact. And then it's going to undergo a change in configuration and sit down. That one cheats because it sits down by twisting its tail fibers, and in actuality the tail fibers bend. But hey, what can we do? It's a model. So this thing has to make contact. One thing about viruses is they tend to be very host specific. As I said, a bacteriophage can't infect you. It's a bacterial virus. Well, how does it know what kind of a cell to infect? And the answer is the cell has to have the right receptors. So in this case, they're showing this virus receptor being a little round ball. It's going to have to go into a receptor that has a site that would fit that little round ball. Only certain viruses can infect certain types of cells, which is why when you think about human diseases that are caused by viruses, some types of viruses infect the nervous system, some infect cardiovascular system, some infect the skin. It has to do with receptor sites on the cell surface. And bacteriophage are extremely host specific. So a colophage is a virus that infects E. coli. A colophage cannot infect Staphylococcus or Streptococcus or Klebsiella or any other type of bacteria. In fact, some bacteriophage are so specific they will only infect a specific strain of Escherichia coli. So again, it has to do with the receptors on the host cell surface. The binding of that virus is very, very specific. With a T-even phage, once all the tail fibers have attached, they flex. Now, as I said, this one they spin in here and cause this thing to sit down. But in reality, the tail fibers flex. This is going to allow the tail to drop, and the tail pins then come in contact with the host cell surface. When the virus is just attached by the tail fibers, apparently it's easy for the virus to get knocked off. Bacteria are relatively motile if they're E. coli, so they can be swimming about in the environment. if they bang that virus into something, if it's just attached by its tail fibers, it'll get knocked off. But once it has flexed the tail fibers and made contact with the tail pins, then it's not going to fall off. It's attached. This business of attaching to a specific receptor and then the flexing of the tail fiber, that's a response. So we said viruses do have some of the characteristics of life. They respond. They respond to a receptor site by attaching and then flexing the tail fibers and sitting down. I guess that's what you call it.
Why would our cells have the
receptors for these? That's a very good question.
Are there beneficial viruses?
So if you think about, this is kind of a loaded question, but think about the function of sexual reproduction, which is to increase genetic variability, right? Mm-hmm. What do viruses do? They increase genetic variability. So is it beneficial? Perhaps. It's you know, sometimes they cause disease and kill the cell, but do they always? I would argue the answer to that question is no. Definitely not. And then think about CRISPR. I hate to keep bringing that up again, but think about CRISPR, Donna, because what's happening is a virus gets into a bacterial cell. The cell takes some of that nucleic acid and incorporates it into its chromosome. And then bacteria can transfer genes to other cells, including ours. So how beneficial is it? We don't even know. I mean, that's a question, like I said, it's a loaded question. I think of viruses as being like this huge post office information exchange service. Sometimes the information you get kills you, but not always. Not always. So there's potential benefits.
And if it doesn't kill you, does it increase your immunity to the next time?
I don't know. It does for bacteria, but does it for us? I don't know. I don't know. What does that information do for us? I don't know. Hard to say. I don't know. And now, now that humans have developed ways of looking at the human genome, they can really analyze and they can begin to say, where did these genes come from? How many of our genes are viral? I don't know. I know that some bacterial ones definitely are. So if theirs are, and they can exchange DNA with us, chances are some of ours are too. So what function do they play? I don't know.
And could some sort of cell therapy be the immunity or cure against viral infections? To not have those host receptor sites?
Yes, definitely. Yeah. So that's one of the targets for preventing HIV, is to block the receptor.
I thought I just thought of that myself right here. Like,
yeah, but you
call
did.
Someone.
You did, yes. And that's exactly right. Yeah. So those are the kinds of things that are happening. But it takes knowledge to understand what's the potential for what can we do. Well, and there's so much we don't know. It's like, we don't know much. When it comes to microorganisms, we don't know much. Anyway, so these guys are responsive. Now, this is talking about, depending on the type of virus, they can get in by more than one means. So could be they're using a receptor that has another function, that they have just evolved a mechanism that allows them to take advantage of it. It's like if you open your door to let glucose in, maybe they sneak in with it. There's different ways they can get in. So this is showing a virus that maybe it could attach to glycolipids, maybe it could attach to protein, sometimes they attach to flagellins, sometimes they attach to pillins. They get in by different means. The other thing is most animal viruses get into our cells because our cells are constantly sampling the environment. Chemicals bind to our cell membrane surfaces and endocytosis happens, so we take them in. So it's like we're constantly sampling the environment. Why would our cells do that? Apparently they're hungry for information. They want to know what's out there. Here, give me some of that.
So I can react appropriately.
Who knows? It's just, I mean, there's this biochemical interaction that's been going on for a very, very, very long time. And here we are now trying to analyze it and say, is this good or bad? But it's kind of like humans looking at all the life on this planet and saying, which ones are good and which ones are bad? Who are we to judge? We have no idea. Good for what? What are we good for? Ask your local whale. They probably would say, not much, actually, not much. Anyway, so this is just showing a salmonella type phage, not a colophage, but a lot of phage can bind different surfaces. Okay, so step one of the life cycle is get attached to the host cell surface. Step two is called penetration. This is going to allow the nucleic acid core of the host to get in. In the discussion of transduction last time, I showed you an image that showed just the nucleic acid core. Again, this part, this is the functional part of the virus that needs to get inside the cell. The rest of this is packaging. It's kind of like when you buy a Twinkie and it's inside a cellophane package inside a box. The box and the cellophane is just packaging. This is the Twinkie. Anybody eat those? Oh, no more Twinkies. Okay, so anyway, penetration is going to allow that nucleic acid core to get inside the host cell. The illustration is showing how a bacteriophage, a T-even bacteriophage attaches. Again, the attachment involves tail fibers first, and the tail fibers flex, and this thing sits down. Then the core of the tail actually shortens, the sheath shortens, which drives the core through the cell surface, and then the nucleic acid goes in. So it's just the nucleic acid going in. This tail assembly, the core surrounded by the sheath, is kind of like a hypodermic syringe. So it's mechanical pushing this thing in, but not all viruses get in that way. So some of them do. Again, tail sheath proteins undergo a molecular change. Here you can see the tail core being injected into the surface of the cell. The sheath shortens, the core goes through, and then the DNA goes into the cell. So all the rest of the virus stays outside. If we're talking about a virus that is infecting human cells, oftentimes you can test to see if you've been infected by a virus by testing for the presence of antibodies. And the antibodies are often responding to the part of the virus that's outside the cell, because that's what they see. The viral nucleic acid gets inside the cell, that's no longer visible to your immune system. So your immune system is looking at the packaging.
So does it just puncture a hole to go in?
Well, the bacteriophage do, but not all viruses do. A lot of them get in by, like a permease can take them in. They get in by different means. Or as I said, animal viruses often via endocytosis. We actually take them in. There's variations on that thing too. So once the virus gets in, and again, this is a summary of virus absorption and penetration, just showing landing, attachment, tail contraction, penetration, and then the DNA going in. After that happens, the virus goes into what's called a latent period. Not surprisingly, a lot of times in life cycles, there's more than one name to the life cycle. As I said, attachment can be called attachment or absorption. Penetration, as far as I know, is always penetration. The next phase is either latent period or eclipse. Latent period, latent suggests that the virus is inactive. That's a misnomer because this is the stage during which the virus is going to accomplish a great deal. Eclipse is probably a better name because eclipse means you can't detect the virus. Again, if this thing lands on the host cell surface, what goes into the cell is this. If this is not a good model for an animal virus, but if this packaging is on the outside of the cell, it's detectable. But once a virus infects a bacterial cell and this thing has gotten in, there's really no way to tell that it's in there until it completes its life cycle. And that's what's a lot of the time for the life cycle is going to happen during this latent period. Because what has to happen is that viral nucleic acid is going to be transcribed. What does that mean? Transcription is RNA synthesis. And the T-even phage is a DNA type virus. So the viral nucleic acid, the DNA gets in, that's going to be transcribed. Initially, that transcription is probably going to involve cellular enzymes. But what happens is the viral nucleic acid will encode new enzymes that will actually be preferentially binding to the viral DNA. So initially transcription involves cellular enzymes and then later on involve viral enzymes. Yeah.
So what if virus is detected but then it makes its way into the cell?
Detected by what?
By the antibodies.
It's too late if it gets in. I mean, other than if you were assessing a patient to see if they had been infected by a virus, then if you're testing for antibodies, you could say, yep, they've been exposed. But then what's going on inside, it depends on, is this virus one that becomes latent and is inactive for a long period of time? That's the other problem with that term, latent period. Latent also refers to a disease category. And some viral diseases are latent, which means the virus is inactive. So eclipse is really a better name for this stage because the virus seems to have disappeared. As a virion, it has disappeared because all that's left of it is nucleic acid. At any rate, so the viral nucleic acid is transcribed, as I said, messenger RNA is going to be made, and then it's translated. Translation involves what structures within the cell? Ribosomes, because translation is what? Protein synthesis. Do not mix those two terms up, please. And on this last quiz, I found out that a lot of people either aren't reading or can't tell the difference between transcription and replication. Transcription is making RNA. You don't have thymine in RNA. I had a lot of people on the quiz making me nucleotide sequences that had thymine in them. And I thought, you're not transcribing, you're replicating. That's not what I'm asking for. So transcription's going to happen, translation's going to happen. The reason that we had to go through all the DNA stuff before we talked about viruses is because you need to know what's going on. The viral nucleic acid gets in, it's going to be transcribed. Again, initially that involves cellular enzymes. Once some of the viral genes are transcribed, they're going to encode enzymes that will preferentially bind to viral DNA and transcribe more viral genes. And once you have messenger RNA, that messenger RNA goes to the host cell ribosome. Viruses aren't cellular, they don't have ribosomes. So they're using the host cell machinery to build the proteins that they need. This is a strange version of a ribosome that is showing the A site and the P site and so forth. Anyway. So again, initially transcription involves viral enzymes, but then after a while, excuse me, it starts out using host cell enzymes. After a while, it uses viral enzymes. But translation is absolutely dependent upon host cell ribosomes because viruses don't have it. Now, there is an exception because sometimes viruses come into cells with proteins inside the capsid. But we'll get to that after a bit. Usually bacteriophages don't. Animal viruses often do. So the viral genome is going to be transcribed. Well, here's another little twist, although not so much, because if you think about what's going on in a cell, not all of cellular DNA is transcribed at once. We've talked about operons, so we know that transcription can happen on a particular operon when sigma factor binds to a promoter, and then you transcribe the genes within the operon. The same kind of thing happens within a virus, so that there are different segments of the viral genome that get transcribed at different times. And they're usually referred to as immediate early phase genes, and delayed early phase genes, and late phase genes. Just keep in mind, they're not all going to be transcribed at once. The immediate early phase genes, these are the ones that are going to be transcribed first, and then again, the messenger RNA is going to go to the ribosome to be translated. These are going to involve making proteins that have a variety of functions. So part of it, again, has to do with getting some enzymes that will preferentially transcribe viral DNA. But the other thing this virus is going to do inside the host cell is destroy the host cell DNA. So we preferentially transcribe viral genes, and then we start making some proteins that chop up the host cell DNA. This is significant because the host cell is going to lose its function then. And the host cell DNA can be used to make viral nucleic acid. We know that viruses don't carry out metabolism on their own, but because they have DNA that encodes proteins, and proteins can be enzymes, and enzymes are catalysts, then we can say, yeah, okay, viruses have the capability of influencing metabolism, because they make a bunch of different enzymes. So another characteristic of life, they are involved in metabolism. They're just going to use the host energy source, the host ATP. Anytime there's synthesis reactions that have to be catalyzed, they're using host ATP to do that. So they're going to break down the host cell DNA, and then they're going to start building virus parts. Some of those proteins that are encoded by immediate early phase genes are regulators. So they're going to regulate expression of genes. We didn't get into all kinds of regulation. We talked about promoter sites and operator sites. We didn't talk about attenuators. And this can be much more complex, so never mind. Some of those proteins are actually going to help to protect the phage DNA. Remember that bacteria have defense mechanisms. They have restriction enzymes. Those restriction endonucleases, which their job apparently is to recognize foreign DNA that gets in and chop it up before it has a chance to take over. So there are going to be some enzymes encoded by the viral genome to help protect them. There's always a contest then between the infector and the infectee. How quickly can the bacterium launch some protection or get protective mechanisms up there? And then the other thing we know is that bacteria will actually steal some of the viral DNA and incorporate it into their chromosome if they can. So when do they get to do that? When are they just killed by the virus? Viruses don't usually kill everybody. Well, the next set of enzymes that are going to be catalyzed or expressed, sorry. The DNA is expressed to make the enzymes. So the next set are referred to as the delayed early phage genes. These are going to encode enzymes that replicate the viral genome. And the replication of the viral genome happens hundreds of times, typically around 200 times. So when we think of a cell reproducing, typically one cell undergoes fission and you get two. Virus replication is completely different because what you do is take your nucleic acid and it's kind of like running a PCR, but it's inside of a cell. The viral nucleic acid is replicated over and over and over again. You get around 200 copies of it, but it's just nucleic acid. Again, it's just the viral genome. This is just showing replication. Now, because mutation happens, and mutation is another characteristic of life, but really mutation is just changing the nucleotide sequence of nucleic acid. So if that viral DNA is being replicated 200 times, and the DNA polymerase that's involved in that doesn't have a really high fidelity, then you're going to get mutations, and you're going to get some changes in base sequence. So not all 200 viral genomes that are synthesized are exactly alike. There's a potential for some change here, which is not too surprising. Then there are genes called late phage genes, and the late phage genes are going to be involved in encoding proteins that are not enzymes, but they're the proteins to make the rest of the virus. So again, if we're talking about a T-even phage, we have the proteins of the capsid, we have the proteins of the tail collar, the sheath, the core, the base plate, the tail fibers, the tail pins, etc. All those proteins are going to be made. If we're talking about an animal virus, sometimes there's enzymes that are incorporated inside the capsid. They would be encoded by late phage genes. So these are just going to come on later. They're going to make a bunch of virus parts. Again, if the DNA was multiplied a couple of hundred times, then slightly different viral genomes are going to be transcribed and translated when we talk about late phage genes, what's being expressed. Maybe slightly different proteins, slightly different amino acid sequences. It turns out it's not terribly critical to the function of the virus because the viruses are mostly dependent upon host cell function. But that protein coat, the capsid, the tail fibers, etc., are going to allow the virus to get in. If you mutate it much, perhaps the virus changes host. Maybe if it was infecting one strain of Escherichia coli, maybe mutations would allow it to infect a different strain of E. coli. So mutations will happen, and as a consequence, these tail components are going to be a little bit different from one another. The next stage is called assembly, and it's exactly that, because the nucleic acid cores are going to be packaged inside these capsids. These things, some of these proteins come together by themselves, others are helped along by proteins called chaperonins. So cells often have chaperonin proteins, and chaperonin proteins kind of bring other proteins and put them together. So these things come together. We build the capsid, we build a bunch of tail assemblies. As I said, tail assembly is a very appropriate name because that's exactly what they are. They're built. Viruses are put together like a bicycle on an assembly line. You've got a bunch of nucleic acids, wrap those up in capsid, so the triangular pieces come together and build the capsid. Then the tails and tail fibers and so forth get put on there. There's chaperonin spelled for you in case you want to see that. Again, chaperonin proteins are sometimes involved, sometimes not. Once the assembly has happened, and you have hundreds of new virions inside the host cell, the next thing that has to happen is they're going to get out. Again, that's the virus strategy. Get in, reproduce. The get out phase, oftentimes this involves enzymes. Basically, they're going to soften up the cell wall, the cell's going to break open, and the viruses are going to get out. So, release or liberation. Bunch of new virions are free, and they're going to go out and infect a new cell. The only thing that comes close to this is schizogony, multiple fission, where one plasmodium gets into a red blood cell and reproduces many times over, so that when that cell blows up, a bunch get out. But it's not hundreds. It's nowhere near the numbers that we're getting with viruses. So we're going to look at a virus life cycle in class, and we're going to see that the rate at which virus particles increase in number is extremely high because of the way they reproduce. So again, here's a summary of the cytolytic virus life cycle. And I showed you this last time when I was talking about transduction. If transduction is generalized, go back to that, generalized transduction means that any piece of the viral genome, any piece of the host cell genome can be incorporated with the virus when it gets packaged. Well, this is because the viral genome comes in. So here's our virus landing. Here's the genome coming in. The host cell DNA is chopped up. The viral DNA is replicated. The tail fibers are, this is capsid being built, and then tail fibers are built. Here's assembly, they're all being put together. When assembly occurs, some host cell DNA can be packaged with the viral DNA inside the capsid, and then when that virus gets out, the next time that virus attaches to a host, that nucleic acid has to go into the chromosome and become a prophage, if you want to accomplish generalized transduction. Make sense? Kind of? I don't know. Have you looked at those notes since last time? Maybe not. Well, over the weekend, that'll make more sense. How long does this process take? Usually a matter of minutes, which is not surprising because Escherichia coli can undergo fission every 20 minutes, which means they're replicating their nucleic acid pretty rapidly. The viral nucleic acid is smaller, typically, than the bacterial genome, actually a lot smaller. So they're going to replicate the nucleic acid pretty fast. How fast the building of the proteins takes place probably depends on the metabolism of the cell. So we're going to be working with viruses in the laboratory. I think the T-even phage life cycle is maybe 30 minutes, 20 or 30 minutes. It's pretty quick. There's different ones. So again, bacteria undergo fission every 20 minutes. It's not surprising that the bacteriophage can reproduce pretty quickly also. A single virus particle might cause destruction of an entire population, but it probably won't. So one virus gets in, if it reproduces a couple of hundred times, a couple of hundred virus particles get out within 20 minutes. Depends on how many cells were introduced into the population to begin with. Remember lag phase, you have a certain number that got in there. Could the virus reproduce so fast that it would infect all of those cells and kill them? Yes, it's possible, but not usually. Usually the bacteria, some of them survive, and that's partly because bacteria have restriction enzymes. They can defend themselves. And partly because they have a CRISPR. They can defend themselves. This is kind of an interesting twist. Because cytolytic viruses are very host specific, and people recognize that some of the bacteria that infect humans could potentially be killed by viruses, they actually use viruses in some countries as a way of controlling bacteria. So in some countries, when infants are born, they're given a little squirt of bacteriophage. Not one type, but kind of a cocktail of bacteriophage. You get a squirt in the mouth. And the idea would be that those bacteriophage can pass into the gut and potentially kill infectious bacteria that might be introduced into that area. How many of you are readers of novels? Okay, so the novel Aerosmith by Sinclair Lewis. Have you heard of it? Play the author's game. You've heard of Aerosmith by Sinclair Lewis. Well, Aerosmith is a story about a physician who is studying plague, and he thinks plague could be controlled using bacteriophage. So it's kind of an interesting, you read that, and it's like, oh yeah, application of just this, using bacteriophage to control Yersinia pestis. Unfortunately, one of the main characters picks up a cigarette that has been infested with Yersinia and infects herself with bacteriophage. Excuse me, infects herself with plague and dies. But oh well. Spoiler alert. It wasn't the main character. It was just a sort of a main character. Anyway, because a lot of places antibiotics are not as readily available as they are here, so bacteriophage are really easy to grow. You can get a lot of bacteriophage very quickly in an artificial environment. If the only hazard is you have to grow the bacteria that you want to get the bacteriophage to infect. So you do have to grow those guys up and then get the phage. But it is used in countries other than this one. This is looking at this kind of application of bacteriophage. Okay, so that's the cytolytic bacteriophage. The next variation on the theme is the temperate bacteriophage. And as I said, temperate, also called avirulent. A means without. So if it's without virulence, again, you have to think from a bacterial perspective. That means it's not going to cause any damage to the bacterial cell. It's avirulent. It's not going to cause disease to the bacteria, or asymptomatic. The bacteria is not going to have any symptoms of phage infection. At least not right away. And I have to qualify that because these phage can revert back and forth. They can be temperate for a while, and then they can be virulent, cytolytic. So they don't always stay the same way. If you have a temperate phage, very often that temperate phage will have inserted itself in the host cell chromosome. If it's in the host cell chromosome, then it is a prophage. Prophage just means the viral nucleic acid is in the host chromosome, and if it's there, then it's going to be reproduced right along with the viral nucleic acid. We now know that this has happened to quite a number of bacteria. We know that they are carrying viral genes, and they are expressing those viral genes. Again, here's a quick review of transduction. Now that we have a little bit better knowledge of viruses. So transduction, transfer of DNA from a donor to a recipient by means of a virus. Transduction is generalized if the virus involved was initially cytolytic. Because it got in, it started chopping up the host DNA, any piece of that host DNA then could be incorporated with the viral nucleic acid inside the capsid. So you just chop up the DNA, you get lots of little pieces. There you see a bunch of little pieces of viral nucleic acid plus host nucleic acid. Some of those get combined and packaged during assembly inside the same capsid. Any portion of the host genome can be involved. But once that virus is released from the original host and goes into the second host, it must become a prophage. Think about that. Why? Transduction is horizontal gene transfer. It's genetic exchange. The why is the same as why does transformation have to be transfer of DNA from a dead donor to a live recipient and not the reverse? If you transfer the virus, if you transfer host DNA into a new host cell by a virus that's cytolytic, it's just going to kill that new host and you haven't accomplished anything. In order for it to be a successful gene transfer, the virus has to become a prophage in the second cell. Once it does that, it goes into the host chromosome, and now the host cell is recombinant. It has chromosomal DNA that is its own, it has some viral DNA in its chromosome, and it also has genes from the original host. So that's generalized transduction. This is talking about a particular phage called phage P1 causing generalized transduction. And it's just showing the donor bacterium. In the first place, it has a dark blue chromosome. That chromosome gets chopped up. Some of it gets packaged inside the viral, the phage particles. They infect. And then in our final view here, The recipient cell has a light blue chromosome, but now it also has some of the chromosome from this host. So this cell is now recombinant. And we had successful generalized transduction. Any piece of host cell chromosome could have been transferred. If transduction is specialized, the virus has to be a prophage in the first place, which means it's in the chromosome, and then only the DNA that's right next to that virus can be transferred. These prophage DNA can't just randomly go into the chromosome. They're going to go in at a specific location, and whatever genes are right next to it on either side are the genes that can be transferred. The other trick to this is it starts out as a prophage, but it has to revert to a lytic cycle. That's how it gets out. So it has to become cytolytic, kill the host. The assembly has to happen where some of the viral nucleic acid and some of the host cell nucleic acid get packaged up together in the same capsid, goes off and infects a new cell, and again in the new cell, it has to become a prophage again. This one, it's even harder to see the DNA that was transferred. There's a tiny little red spot over here. Presumably it's nucleic acid from the original host. Anyway, difference between generalized transduction and specialized transduction is which DNA can be transferred and what is the status of the virus to start with. If it's generalized, the virus is cytolytic to start with. If it's specialized, the virus is a prophage to start with. In either case, they have to become a prophage when they get into the second host. Otherwise, the cell's just going to die, and you haven't accomplished anything. So this mentions bacteriophage lambda. Bacteriophage lambda I talked about a little bit last time. I showed you diagrammatic representation of bacteriophage lambda genome. It looks like a closed loop, but it's not covalently closed. It's closed by cohesive termini. So those pieces of DNA are actually linear, and they're just held together by overlapping ends. Bacteriophage lambda is often temperate, which means bacteriophage lambda will typically go into a host cell chromosome and become a prophage. But it can also revert to a lytic cycle. Because it's a cophage, and because people can so easily grow E. coli in the laboratory, they know quite a bit about bacteriophage lambda. Does anybody, well, no, we haven't done it yet. Tonight we're going to do gel electrophoresis, and we are going to use a standard, kind of a calibration standard that shows a set of specific sizes of DNA fragments. And what we use is the genome from bacteriophage lambda that's been cut with the restriction enzyme ND3. I drew that pattern for you guys on the board. Like, oh yeah, that was last time. I'll do it again. This is what bacteriophage lambda looks like. So it has a tail assembly. By the way, the symbol for bacteriophage is phi, and I typically draw it like this. I guess this is the cursive version, because otherwise it's supposed to be like this. But anyway, and this symbol is lambda. So this is bacteriophage lambda. Just a Greek letter. So this is a double-stranded DNA type virus. It has a genome that's double-stranded DNA, which makes sense. That's why it can incorporate into the host cell chromosome. Again, that genome is not really covalently closed circular DNA. It's linear DNA, where the two strands are held together by these overlapping cohesive termini. When talking about restriction enzymes and how they cut the nucleic acid, remember I said sometimes they cut and form cohesive termini. So the example that I gave you, EcoR1 yielded 4 base cohesive termini. This is much longer than that. Again, what makes this cohesive? Those are going to stick to one another. They stick how? Hydrogen bonding. Anytime you have complementary bases, that's how they stick together. Hydrogen bonding. They stick to each other. That's what holds this loop in a loop. Otherwise it isn't really. Because it isn't really a loop, it doesn't require special enzymes to release or to cut it and allow it to go into the chromosome, those cohesive termini just come apart. And then this thing can insert into the chromosome. But again, it gets in at a specific location. It can't just get in any place. I did mention last time that cloning vectors can be made from bacteriophage lambda. If you remove the lytic genes, you have a pretty large loop of DNA that can be genetically modified. Pieces can be put in there. These cloning vectors that are made from bacteriophage lambda are called cosmids, as opposed to plasmids. They're bigger than plasmids by quite a bit. This is a big virus. Well, typically, when bacteriophage lambda goes into a host cell, it doesn't become lytic. Instead, it goes into the chromosome. But because it has a capability of going either way, researchers wanted to study this virus and try to understand how is this controlled. Why does it sometimes go into the chromosomes and other times be lytic? So this mentions a pathway that is lytic or lysogenic. So we have another term we have to define, lysogenic. Lysogeny is the condition that exists when a prophage has gone into the host cell chromosome. So we say that the cell has undergone lysogeny. The cell has also undergone lysogenic conversion. So a lysogenic cell, if an E. coli cell is lysogenic, it means it has a prophage in its chromosome. But now, take that word apart. Lyso means what? As in lysosome, lysozyme, lysis, split. To split. And genic means generating, right? So lysogenic, it sounds like, wait a minute, if that virus, if the cell has a virus in its prophage, the cell is capable of lysis. The terminology came about because people would be growing populations of bacteria and they'd have them in a laboratory and maybe they'd do something to them, like expose them to UV light, and they all died. It's like, wait a minute, they didn't just die, they disintegrated. They were completely taken apart. Other times they'd be growing them in the laboratory, maybe expose them to a chemical, and they'd all just totally die. What they found out is it was because those cells had viruses in their chromosome, they had prophage. And at a certain point, because of stress, the prophage DNA got out and became lytic, and it killed the cell. So these cells were in fact lysogenic. They would generate their own lysis. So that's where the terminology comes from. If they're lysogenic cells, they're carrying a prophage in the chromosome, but that prophage doesn't necessarily stay there. It can get out. Well, so then people wanted to know, what else can happen? If a cell can carry viral DNA in its chromosome, comes back to your question, is that ever beneficial? Well, there are quite a number of bacteria that are recognized to express a particular phenotype that we now know is encoded by viral genes. If a phenotype is changed because a cell has a virus get into it, this is referred to as lysogenic conversion, or it may be called prophage conversion. So if you have a population of bacteria and they don't have a virus infecting them, and suddenly they get a virus infecting them, they'll take on a new phenotype. This new phenotype is the result of lysogenic conversion, or prophage conversion. It means the same thing. They've got a viral nucleic acid in their chromosome. What's kind of interesting is one of the most common features that's encoded by these viral genes is toxin production. So we've been introduced to a number of potential pathogens in the genus Clostridium, Clostridium tetani, Clostridium botulinum. These are bacteria that produce some of the most potent exotoxins recognized, and those toxins are encoded by viral genes. There are toxins associated with many other types of bacteria where those toxins are encoded by viral genes. So is that beneficial to the bacterium? I don't know. Is it beneficial to them if they kill their host? Maybe they have a really good food supply, maybe for a short period of time. But toxin production is the most common characteristic, which then takes us back to the name virus. The Latin term means toxin. So it's not too, we're not too far off in terms of the naming. So these viral genes that end up being incorporated into bacterial chromosomes often encode toxin production. So, term virus, poison. Well, let's back up a bit. This bacterium that's carrying a prophage, again, it's called a lysogenic cell, and people wanted to know what controls whether the virus stays a prophage or gets out and becomes something else. And it turned out it was kind of a combination. It's a control that's exerted by the virus and by the cell. Again, it makes us wonder, where did viral nucleic acid come from in the first place? So here we see a bunch of bacteria. This is talking about bacteria seeming to initiate their own lysis. They didn't really initiate their own lysis. It was the virus that did it. If you're growing lysogenic bacteria in vitro and you expose them to ultraviolet light or toxin, they die. The whole population dies because the virus comes out of the chromosome and becomes cytolytic. Well, it turns out when you look at the bacteriophage lambda genome, there's an operon associated with the lytic gene. So there's a gene in the viral genome called C1 that encodes a repressor protein. And we know what repressor proteins do, right? Repressors bind to DNA at a site called the operator. If a repressor binds to an operator, then whatever structural genes are beyond that cannot be expressed. And apparently what happens when bacteriophage lambda goes into the cell, normally it expresses that repressor protein. So that is a constitutive repressor. All the repressors we've talked about thus far have been constitutive. The virus gets into the host cell, the repressor protein is transcribed and translated, the repressor gene is transcribed and translated. That repressor binds to the operator that controls the lytic genes, and they can't be expressed. So then the virus is going to stay in the chromosome. It's going to be a prophage. It's going to stay put. Whether it stays in there or not is dependent upon the host. So bacteria, Escherichia coli, have a gene called REC-A. And the REC-A encodes a protein called the REC-A protein that has a number of functions, but in association with the bacteriophage lambda life cycle, that REC-A protein is proteolytic. So if the cell expresses REC-A, that protein will destroy the repressor protein. This is inactivation of a repressor that doesn't involve a co-repressor binding to it, doesn't involve an inducer binding to it. It involves that repressor being destroyed. So an enzyme takes it apart. The REC-A protein destroys that repressor, and once the repressor is destroyed, then the lytic genes can be expressed. Well, so then the question is, what causes REC-A to be expressed in E. coli? And the answer is stress. You can stress them with ultraviolet light, you can stress them with toxin, you can probably stress them in a variety of ways, but apparently REC-A, that gene is activated when the bacteria are stressed. So it's kind of like the bacteria are stressed, they're experiencing a tough situation, they express an enzyme that destroys the repressor, which is blocking the transcription of the viral lytic genes. Those lytic genes become expressed, the virus goes through and completes its lytic cycle, gets packaged up as new virions, which get thrown out of the cell. It's kind of like, this ship is sinking, take our genetic information and throw it out in bottles into the ocean that can be picked up by some other cell at some other point. So where did the viruses come from in the first place? Did they come from the cell and get out of that cell because the cell was dying and that information had to go off and get incorporated someplace else? I don't know. At least some of it does now. So if an E. coli cell is dying and that E. coli cell is carrying bacteriophage lambda is going to go back into a lytic cycle and get out. The cell was going to die anyway, so it doesn't matter that it kills it on its way. There you see an illustrated version of a cell blowing up. The cell's nucleic acid is destroyed and viruses are getting out. Oh my goodness, there's a lot of things happening there. This is supposed to be showing lysogeny, and it's supposed to be showing a lytic cycle where the virus just keeps infecting the cell and getting out, and then it's showing lysogeny. In this case, here's phage getting in, nucleic acid comes in, the virus is completing its life cycle, blowing up, and new virions are getting out, which then start that whole pathway over again. But if you go to putting the virus DNA in, here's the viral DNA, now it's in the chromosome as a prophage. If that cell is just reproducing itself, carrying that viral gene, or set of viral genes, it's a lysogenic cell, and this is a condition known as lysogeny. And that can just continue to be reproduced. Here you just see a whole bunch of cells being reproduced. They're all carrying the viral gene. How long do they stay in there? Perhaps indefinitely. Especially since now we're finding these genes associated with bacteria where it was assumed they're bacterial characteristics. They're not, they're viral characteristics. This last little bit has to do with viral classification. I will never ask you this stuff on an exam, but it's just kind of fun to look at it and think about viruses. So again, viruses were initially classified based on what kind of organism they infected. Were they animal viruses, plant viruses, bacteriophage, what have you? And so a bunch of different people were studying them using lots of different terminology and lots of different criteria. Eventually, there were people that called themselves virologists, and virologists said, okay, look, we don't like that the botanists have a bunch of different terms and the zoologists have a bunch of different terms and the bacteria people have a bunch of different terms. We want to talk about viruses, and we want some consistent terminology, especially with viral classification. So this is showing you some examples, viruses that infected tobacco plants and caused specific characteristic infection were called tobacco mosaic virus. We had barley yellow dwarf virus, apple chlorotic leaf spot virus. Had to do with what did they infect and what kind of symptoms. With animal viruses, this became very problematic because animal viruses can often infect more than one host. For example, the viruses that cause influenza in humans can infect humans and waterfowl and pigs. So how do you name that? In us, it causes influenza. I'm not sure what it does to waterfowl. We do have human wart viruses. We have other viruses that are more specific. Of course, rabies virus infects specifically mammals. But around 1960, the virologists proposed that viruses should be classified according to some other scheme, not classified based on what they infect, but classified based on their shared properties. And what they said was they all have to have nucleic acid, which was true, but some had DNA, some had RNA, some had double-stranded, some had single-stranded. They said, what do they all have to have in order to form their protein coat? A protein coat, the capsid, is an essential component of all viruses. Their nucleic acid cores vary a lot, but they always have to make a protein coat. Therefore, they must all make messenger RNA. And they said, let's classify them based on how they get to messenger RNA. And this became known as the Baltimore classification system. It was proposed by David Baltimore. And it puts viruses together in a whole bunch of different categories based on how they get to messenger RNA. What you run into in this system is two different categories of DNA, plus strand and minus strand, or positive strand, negative strand, which can get confusing. But if you just remember that messenger RNA is always positive, then the same nucleic acid as messenger RNA is positive strand, and the complement to that is negative. If a cell has, well, cells always have double-stranded DNA. It's the negative strand DNA that's going to encode messenger RNA. But viruses can have either, or sometimes both. So they looked at the nature of nucleic acid, they looked at the symmetry of the viral capsid, whether they had an envelope or not, dimensions of the viral capsid, etc. But again, the Baltimore classification system is based on how do they get to messenger RNA? How do they do that? So here's some variations. This was developed in 1971. It has to do with their strategy for gene expression. And it's just kind of interesting to see the variations on a theme. Again, you really have to have a pretty good handle of what we're talking about with gene expression, transcription. And then we're going to talk about reverse transcription after a bit. So messenger RNA is positive sense, or positive strand, and DNA that has that same sequence is positive sense or positive strand. Therefore, the strand that encodes that, which is the complement, is negative. And again, you're not going to get this on test. So typically, this is what happens within cells. Cells have DNA. DNA replicates to yield more DNA. DNA is transcribed to yield RNA. And if that RNA is messenger RNA, it's translated to make protein. So if I put this on a quiz, would you be able to tell me what's happening at the arrows? DNA to DNA is replication. DNA to RNA is transcription. RNA to protein, translation. You need to know that. Absolutely. Hard and fast. Don't be confused by those terms. So, viral DNA can come in as single-stranded DNA. If it's negative strand, single-stranded DNA, negative strand is complementary to messenger RNA, so it can just code for messenger RNA directly. If they come in as single-stranded DNA, but it's positive strand, which is the same sense as messenger RNA, they can't encode messenger RNA directly. Instead, they came in as positive. The positive has to encode negative, which then can encode messenger RNA. So there's two possibilities right there. If it's double-stranded RNA, it's going to behave just like cellular DNA. The two strands are going to come apart, and the negative strand is going to encode messenger RNA. If they come in as single-stranded RNA and it's positive strand, they can just go directly to the ribosome and be translated. So some viruses, the nucleic acid that comes in goes immediately to the ribosome. It's positive strand RNA, and it's translatable. So this tells us also, where do viruses go when they get into a cell? Some of them have to go into the nucleus, but some of them don't. It depends on what kind of virus they are and what kind of cell they're infecting. If the viral RNA is single-stranded RNA and it's negative strand, it has to be transcribed, or actually, no, it's not transcribed, because transcription is copying information from DNA into RNA. If it's already RNA and it's encoding another RNA, what process is that? It's RNA replication. Oh, man. So, yeah. Anyway, so it has to replicate, make the negative strand RNA, which is then translated. Oh, and if it comes in as single-stranded RNA that is positive strand, it can either be replicated to yield a negative strand, or, and why would they do that, but they do, they come in as positive strands. Sometimes they go directly to the ribosome, sometimes they don't. Sometimes they will code for a negative strand, which then codes for another positive strand that goes to the ribosome. Or they can come in as negative strand RNA, which then encodes DNA. For a long time, this was just against all rules of biology. Biologists said, no, the information chain is DNA encodes RNA, encodes protein. You can't go backward on that chain. And it turns out they were very, very wrong, because there are viruses that can reverse transcribe. So when I was talking about polymerase, DNA polymerase, is it DNA-dependent DNA polymerase, or is it RNA-dependent DNA polymerase? Cells contain DNA-dependent DNA polymerase, but viruses can bring into cells RNA-dependent DNA polymerase. That enzyme is called reverse transcriptase, and it will reverse transcribe information on RNA into DNA. It also then replicates the DNA and makes it a double strand. So lots of variations on a theme. Again, you're not going to be tested on this. We will talk about reverse transcription more because we're going to talk about HIV, but that's next time. And so the test ends with bacteriophage. And we'll do animal viruses next time. Yep. We'll see more about that next time. Right on. Are we still behind one lecture? I think so. I think we are.
It says November 2nd, human viruses and viral diseases. Yeah.
Yeah, we're behind. Yeah. Okay, so we're still behind in lecture, but not terribly behind. Still behind.
human viruses.
Okay, so did everybody see the message on the webpage about what the lab quiz is about?
No.
No.
No one saw it.
Yeah,
I saw it.
When did you put it up?
I think it was Tuesday because Rosie reminded me.
No, I
Yes,
didn't.
she
No,
did.
I didn't.
What's on the lab quiz?
I did
And
not
sent
say
her an
anything.
email telling her, and then I said, I
I'll
gotta
hold
post it.
out. Damn it. No one's listening.
It's still there.
She
Yes,
posted
that's
it
good.
one
All right. Yay. That might be the last one. Then I can take that stuff off.
yesterday.
Yeah, that's I think you're right. There's still some soil ones that other people have done. You're not all three of them are right there. Okay.
You put that
The stuff my kids really taking no heed in.
the laboratory quiz for today.
No.
Better PCR.
It did say Thursday, and I changed it to today, because it said Thursday.
Oh.
I'm gonna put it in the can. I just always, yeah. Yeah, it was a.